Hello, everyone. Thank you all for joining us for our March Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsors for this webinar, Neurocrine Biosciences and Wave Life Sciences. Also, thank you to everyone who supports the HDF. Because of you, we're able to make the research possible that we're featuring today. And thank you all for tuning in. So I am excited about today's webinar because I get to introduce you all to three amazing HDF-funded HD researchers who also happen to be editors at HD Buzz. If you don't know, which hopefully everyone on the call does, if you don't know, HD Buzz is an HD research news website written in plain language by scientists for the HD community. So I am very honored to be able to work with all of these fantastic people in that capacity. We are joined today by Doctors Rachel Harding, Tam Maiuri, and Jeff Carroll. Rachel is an assistant professor at the Department of Pharmacology and Toxicology and a principal investigator at the Structural Genomics Consortium in Toronto, Canada. The focus of Rachel's research is the Huntington protein, which is expanded in people with Huntington's disease. Rachel studies the structure and function of the Huntington protein in both its unexpanded and expanded forms, with a view to better understand the mechanisms of disease, as well as trying to find new avenues of therapeutic intervention. Tam is a research associate in Dr. Ray Truant's group at McMaster University in Canada. Her work in the Truant lab focuses on the normal biological functions of the Huntington protein in the hopes of understanding how they may be disrupted upon inheritance of the expanded Huntington gene that causes HD. Jeff is an associate professor in the Department of Neurology at the University of Washington. His scientific interest is in understanding the systemic effects of Huntington's disease, especially metabolic changes, which he studies using a mouse knock-in model of Huntington's. He also has an interest in gene silencing approaches and HD therapeutics, particularly using antisense oligonucleotides. We are very excited to have you all with us today, so thank you so much for joining us to share your work. Before we start, I want to remind our audience that you can ask questions in the Q&A box throughout the talk. So the Q&A box, not the chat box. Feel free to ask whatever question might come to your mind, both technical, non-technical. We should hopefully have about 15-ish minutes at the end for a Q&A with our speakers. So send your questions in as you think of them so that you don't forget. And we will try to get to as many of those as we can. I also want to remind everyone this will be recorded and available on our website and YouTube in about a week or two. So if you want to watch it again, or if you want to share it with someone who can't be here, it will be available for that. Thank you again to everyone for being here, and I will pass it over to Rachel.
Hopefully you can all see my slides now and hear me clearly and see me clearly. I first of all just want to start off with a big thanks to the HDF for inviting me to talk to you today. It's such an honor and a privilege to be able to speak to people who are interested in the work that the HDF does and help fund it. So thank you very much for that. So yeah, as Sarah mentioned, I'm actually based at the University of Toronto in Canada, where I work for an organization called the Structural Genomics Consortium, or the SGC. But I didn't start off there. As you might be able to tell, I'm not Canadian, and actually grew up in the UK, in my hometown of Dover, where we have lovely white cliffs and beautiful seaside. But I left there to move to Oxford, where I did both my undergraduate and my DPhil studies. And DPhil is just a funny word that people in Oxford use for a PhD. But I did all my training was in biochemistry, protein biochemistry specifically, and trying to understand how protein molecules work and what they look like by using special methods that we call structural biology. And from there, I moved from the spires of Oxford to, cosmopolitan Canada in Toronto. So I did my postdoctoral fellowship at the SGC, actually, and initially had planned to come for just a year or two, and I'm still there almost 10 years later. So that tells you what an awesome place Toronto is, and I highly recommend anyone to go and visit and see all the things that are happening. So what we do at the SGC is what we like to think of as pre-competitive drug discovery. So this means we're right at the very early stages of thinking about what different things might we want to target with drugs in the future to treat all kinds of different illnesses. And we do all of that in the open as well. So what that means is that we're not filing patents. We're not trying to make money out of this. We're just trying to get things done more quickly, because as we all know, there are many diseases, Huntington's included, where we really need to speed up and accelerate the way that we're making drugs. And we think that by sharing things earlier, we can probably get that done faster through lots and lots of different collaborations. So we're actually a global public-private partnership. So we work with pharmaceutical companies, biotechs, other industry partners, and we have sites all over the world. So I'm based in Toronto, but we also have sites in the US at UNC Chapel Hill. We also have a site in Germany, one in London, all over the place. But, oh, there we go. It's actually the site in Toronto where most of our Huntington's research is based, and that's the team that I lead that you can see here. And this is like a really cool group of people, all of whom are extremely dedicated to understanding Huntington's disease, and they're a very talented bunch. Most of the work I'm going to talk to you about today has been done by Renu Chandrasekhan, Manisha Yadav, and Cheryl Arrowsmith. And this has all been funded by the HDF and some other patient organizations. So what we're really interested in is thinking about biology right at the molecular level. So we can study Huntington's disease, for example, in the case of humans, or maybe we just think about the brain itself or nerve cells. But what we like to think about is going all the way down to think about single molecules and how they are working, understanding these kind of molecular machines, to say. And this is because proteins, I would argue, are some of the most exciting and important molecules in the cells of our bodies. So these are the molecules that do all the stuff that make us work and make us function as living beings. So we have proteins that most of you probably know about, things like keratin that's in your hair and your nails, things like hormones and insulin, that's a protein. All of our immune response is, orchestrated through antibodies and other proteins. And then the way that we digest our food is also through enzymes, such things like amylase. But as a structural biologist, what that really means is what I like to do is understand how proteins work by looking at their three-dimensional structure. And we can kind of think about this with looking at two tools that I've pulled out of the toolbox here, which both have a very similar shape. But we just looking at these shapes covered with the blue and green boxes is not necessarily clear what these two tools do. And it's only when I reveal the boxes and show you that these two different tools, you can see we've got some things which are for plying and others that are for cutting. We can see that they actually do two different jobs. And this is kind of what we want to do with protein molecules. If we can really see exactly how all of the atoms that make up each protein molecule are connected and organized in three-dimensional space, then we can actually try to understand how they're actually working in cells. And so we wanted to apply this to the Huntington protein. So most people on this call probably already know Huntington's disease is caused by this mutation or expansion in the Huntington gene itself. But the Huntington gene encodes for the Huntington protein. So when you make your protein, you'll have regular protein that doesn't have the expansion, but people with Huntington's disease will make a slightly different version of the protein that has this expanded region at the very beginning. And so what we wanted to do is try and understand, let's look at these proteins and see what's going on. What can looking at the protein tell us? Before we look at the protein, we've actually got to be able to make it. And so one of the first things that we did with this very talented team here, Alma Saitova and Peter Lopnau, was we first took the gene or the piece of DNA that encodes for the Huntington protein, and we actually put that inside insect cells, which sounds really, really weird, but it actually turns out that insect cells are really, really great at making human Huntington protein. And we grow these cells not as insects flying around the lab, but actually in these big buckets that you can see in this incubator here. And inside these huge buckets, we can actually make lots and lots and lots of Huntington protein by the bucket load. And we can make Huntington that looks like Huntington from people who don't have Huntington's disease, but also people who do. And then what we can do is we can cross compare and see how those might look different. So because we developed all these tools to do all this stuff, we have this open science remit, we didn't want to just keep all these tools to ourselves. We actually shared them with people all around the world. And so any researcher anywhere can actually access all these pieces of DNA that we made. And actually, I have a lot of people emailing me, asking me to send them bits of the Huntington protein as well. And we've shared samples with, all kinds of people all around the world, all with the aim to try and accelerate research. So this is what the Huntington protein looks like. And it's this big kind of cobbled mass. And you can see all of these kind of nooks and crannies and other bits and pieces. And we did this using like a very high-powered specialist microscope. This kind of builds upon work that other people have done looking at Huntington. But so far we have one of the most detailed insights into exactly what's going on. And what structural biologists call this is they call it atomic resolution. So it means we know where every single atom is, which is really exciting for someone like me. And when we have this kind of information, we can start to think about, okay, well, what's going on? What is Huntington doing? And how does this change in disease? And one of the first things we want to look at is, well, what does Huntington hang out with? Most molecules in the cells of our bodies don't exist in isolation. They're always hanging out with other partners and other molecules that they work together with to do their jobs. So we wanted to try and work out, well, what's Huntington hanging out with in cells? And when we look at our model, because we've got this level of detail, we can start to think about, well, what does the surface of Huntington look like? Is it greasy? Is it sticky? Are there charge patches? And it turns out there's a lot of different things going on. But in particular, what we noticed was there's this really sticky patch of positive charge found on the surface of the Huntington protein. And we know that things that are positive like to stick to things that are negative. And we'd also at the same time noticed that when we made our Huntington protein, we were actually pulling down message molecules, or these RNA molecules, which are very negatively charged. So we started to think about, are these two things sticking together? And is Huntington binding RNA message molecules? And so we worked together with this really awesome Canadian team of scientists. So Christopher Pearson, Hanson Hay, and Mahmoud Pouladi, all of whom are based in Canada, in Toronto, and in Vancouver. And we were able to figure out that actually, yes, Huntington is binding onto these RNA message molecules. And in particular, there was one RNA message called NEAT1, which was coming down every single time. Like we always saw this with Huntington in our studies, which is rather exciting and pretty neat. And then we did some really cool imaging. So this is where we look to see, okay, well, when you take Huntington out of cells, we can see this NEAT1, but let's look back in cells. And so then with this, we use microscopes to look inside cells and look specifically at where the Huntington molecules are and where the NEAT1 molecules are. And so the Huntington here is shown in green, the NEAT1 is shown in red. And when they sit on top of each other, we actually get this yellow signal in our microscopes. So this is really exciting because it means that we can see these two things in the same space within the cell. But we want to get into more detail of that. How are these two molecules sticking together? And then with another awesome Canadian team, this is not an advert for Canada, by the way. We have all of these folks who are listed on the slides are Tam, Ray, Sarah, and Sam, who are doing all of these different ways that we can try and look at how these NEAT1 molecules and the Huntington molecules stick together. And we're getting lots of really exciting results. So you might ask, okay, well, this is all great, Rachel, but what does this mean and why does it matter? And I would argue that it's really important that we understand what the normal Huntington protein is doing in our cells, because if we understand what it does normally, we can maybe think about how this changes in Huntington's disease. And this kind of information is really useful to help us guide the development of new medicines that might be able to treat Huntington's. So I'd just like to end again with a big thank you to the Hereditary Disease Foundation for all of their support of this work and all of the wonderful people who have helped us along the way. Science is a big collaboration, and we've been very lucky to work with some really awesome partners to get all this stuff figured out. Okay, thank you.
So thanks so much for having me to talk about what we're working on in the Truant Lab. It's really a pleasure to join in this talk with friends and colleagues. So I'm going to be focusing on how we're targeting DNA damage in Huntington's disease. But I'm going to start with a bit about my HD family history. So I imagine a lot of people on the call will be able to relate to this story, but basically the first that we knew about HD in our family comes from my great-grandfather, Roy Smith, who had the gene and passed it on to all 4 of his kids, one of which was my grandfather, Victor. And it was in that generation that we learned about HD. His brother was diagnosed by a doctor in Toronto, actually. And so we always knew that my mom had a 50-50 chance of inheriting the gene. And unfortunately, she did inherit it, and we lost her a few months ago. So I just wanted to dedicate a slide to my awesome mom, who is also an awesome grandmother. this is what brought me to the field. So it was around the time that I was finishing my PhD, which I actually did in cancer research, that we learned that mom carried the gene. And so I switched fields and looked for the closest lab that was working on HD, which happened to be Ray Truant's lab at McMaster. So what we do in the Truant lab, as Sarah explained, is we try to understand what the normal Huntington protein is doing in cells, in hopes of understanding what might be going wrong when it has that expanded bit that Rachel was explaining. And so the basic research that I've been focusing on in the Truant lab is on the process of DNA repair and Huntington's role. And so I always love to show this image because it kind of represents one of the most exciting moments in my scientific career. There's something really special about when you know something, when you're seeing something that nobody else in the world knows. And that was what was happening at this moment. So what we're looking at here is cells grown in a dish. And we can actually damage the DNA using a laser and then kind of let the cells recover, and any proteins that are involved in DNA repair will usually move to the sites of damage. And so it was quite surprising when we saw that the Huntington protein was there. We also, went on to find that the Huntington protein, like Rachel's question, who is Huntington hanging out with? We asked that question in the context of DNA damage. So under DNA damaging conditions, what proteins is Huntington hanging out with? And we found that it was interacting with DNA repair proteins. So this is just an illustration to give us an idea of what this might look like. This isn't actual data or anything, but it's just to show you what it might look like when DNA repair proteins are going to the DNA. And we know that Huntington interacts with these DNA repair proteins. We also know that it's interacting at least on this very smooth surface called mica. We know that Huntington can interact directly with the DNA. So this is similar to the data that Rachel was showing you. It's called atomic force microscopy. And the way that we figure this out, it's very cool. It's kind of like a record player going over the grooves on a vinyl, but it's like a super, small type of needle that's going over the molecules and bumping over them and able to generate this image. So you can see the little Huntington proteins, and this string is a piece of DNA, and the Huntingtons all lined up like beads on a string. And so this was very exciting. And, when we made this initial finding was several years ago now. And when we made that finding, we were maybe a little bit naive in understanding like the links between DNA repair and neurodegenerative diseases. But as soon as we looked into the literature, we found that other neurodegenerative diseases are also linked to DNA damage and DNA repair. So some of these diseases are inherited. In fact, a lot of neurodegenerative diseases, genetic ones, are caused by a mutation in a DNA repair gene. Others, like Alzheimer's and Parkinson's, also have environmental factors. And then over the years, lots of evidence of DNA damage in HD patient samples and mouse models and cellular models has built up. But probably our biggest clue comes from large human genetic studies that have informed us that DNA handling and repair mechanisms are actually influencing age of onset. So whether people start to develop some symptoms early in life or late in life. And those who are familiar with HD research will know that most labs are focusing on a very specific aspect that was revealed by these large studies called GWAS studies, which is on the mismatch repair arm of DNA repair. And this is really important because we know that the mismatch repair proteins influence this thing called somatic instability. Somatic instability is when those CAG expansions further expand in some cells in the body. And we know that mismatch repair influences somatic instability. So there are lots and lots of labs now working on this line of investigation, which is awesome. The Truant lab is actually focused on global DNA repair and not just somatic instability. So, it's an understudied thing that we feel is very important because the links between DNA damage and other neurodegenerative diseases that don't have things like somatic instability suggest we really need to follow this lead. So that's what we're doing. And in fact, we have found that one of the proteins that many neurodegenerative diseases converge on is this protein called PARP1. So we started looking at the relationship between PARP1 and HD when we did this study looking at what proteins hang out with Huntington under conditions of DNA damage. And we found that a large proportion of the proteins that were hanging out with Huntington under these conditions were, they carried this molecular tag called poly-ADP ribose, or PAR for short. So PARP1 is the protein that is responsible for generating that tag. It creates these long chains of this PAR tag on proteins that are working to fix DNA. And these PAR chains kind of act like a net to recruit more DNA repair proteins and fix the damage. So the most interesting and relevant thing that we found in collaboration with Lauren Byrne and Ed Wild and tie-in from the Dawson group at Johns Hopkins, we found that the PAR levels were reduced in the spinal fluid from HD patients. And this is happening even before symptom onset. Now, this was very much a surprise because we expected the PAR levels to be higher in HD spinal fluid. That's what happens in most other neurodegenerative diseases, and it makes sense because if you have more DNA damage, you would expect to have more PARP generating the PAR and have all of this PAR. It's not what we saw. It's very interesting, and we went on to find that in cells that we've derived from HD patients, it seems like the PARP reaction is subdued. It's not happening properly. Now, this might be because of a direct interaction between the Huntington protein and PAR itself. So here's another atomic force microscopy image. And so this is what PAR chains look like. And you can see they're about 2 nanometers high. So the white indicates it's coming off the surface or out of the screen. And when you add Huntington to this mix, you can see that the Huntington protein seems to be globbing on to the ends of the PAR chains. And this reaches out off the surface up to 10 nanometers, so much higher. So gathering all of this evidence, and at this point in the project, I guess it would be about a year ago now, funding was running out. It's time to apply for more funding. So that's always a nice time to pause and really ask what's important and what you want to work on. And so I did some soul searching and just asked, what does the field need the most right now? And how can I contribute with what I know how to do? And the 2 most important things that I could come up with were we need early, easily accessible biomarkers to track disease, hopefully before symptom onset. And we need this to help with clinical trials, gauging whether drugs are working or not, how do you tell a drug is working if you're aiming to treat people who don't have any symptoms yet? So we need biomarkers. And although the most promising strategies like Huntington lowering and targeting somatic instability may be going after the root cause of disease, there's still work to be done. It's being done. It's very exciting. And I'm very hopeful for those approaches. But in some cases, they still need development, or they require, crazy administration like brain surgery. And so I think a very important question that we have to ask ourselves is if there are existing drugs out there that may help HD patients. And so we can't ignore the availability of these existing drugs. There are cancer drugs that target PARP. Whether or not that's going to make sense for HD is still very much an open question, but it's something that we're working on in the lab. And this drug is very interesting, omaveloxolone, also known as Skyclarys. That was just approved for Friedreich's ataxia, which is another inherited movement disorder. And so this drug in particular improves mitochondrial function, which if you remember from your high school biology, the mitochondria is the powerhouse of the cell. And we know that there are connections between mitochondrial function and DNA damage. And so these are the kinds of things that I was thinking about, and my goals became to determine whether DNA damage would make a good biomarker, and to generate some preclinical data on some of these pre-existing drugs in the context of HD and see whether they're worth pursuing or not. So I was very excited and thankful when the HDF funded the study for the first question. It's a very straightforward question. Basically, can we detect increased DNA damage in pre-symptomatic gene carriers in an easy access format? So in collaboration with North York General Hospital, the Enroll HD site there, they are collecting finger prick blood samples from our very generous study participants. And what I'm working on in the lab is the best way to easily measure DNA damage in these small volumes of whole blood. So I'm looking at two different approaches right now. I'm not going to go into them for today's talk, but I'm happy to answer any questions if anybody wants to hear me go on about measuring DNA damage. So just to sum up, by the end of this year, we'll know whether or not there's a detectable difference in pre-symptomatic patients in these small volume of blood samples. We will know whether we have a feasible method for measuring DNA damage in these finger prick blood samples, and whether this option is worth pursuing, or whether we should focus our efforts elsewhere. So I'll just end by thanking our funding sources. We can't do any of this stuff without the funding. The Truant Lab is a great group of big brains who are all working very hard on these problems, and we're having a good time doing it too. And I just wanted to mention, Saturday is Huntington Gratitude Day. So this is where we celebrate the partnership between HD families and the scientists that are working so hard to find a treatment. It's long-standing partnerships, and every March 23rd is a day for expressing our gratitude. So if you're on social media, you can share a picture of yourself making the heart with the H sign. All right, thank you.
Myself again. Hi, I'm Jeff Carroll. Very pleased to be here and give a talk about work that's been happening here at UW that was catalyzed by my lab, but mostly it's been happening in a partner lab, as I'll describe here at UW. But I know there's a lot of families and stuff, and so I wanted to also start with how did I get interested in HD research? This is me pointing at myself for some reason. I don't know why. And apologies to my two youngest sisters who, for some reason, maybe they weren't born yet, aren't in this photo. There's actually 6 of us, but I guess it wouldn't all fit on my mom's lap. This is my mom, Cindy Carroll, and she passed away from Huntington's disease. And so for me, growing up, we didn't talk about it. It was kind of a weird, kind of dark secret in the way it is. You know, it shouldn't be like my grandma had Huntington's. In retrospect, it was very obvious, but you know how families are, and I'm sure many families in the audience have similar maddening stories. But anyway, here in happier times, here's my mom, who loved being a mom and was incredible with kids, and she had 6 kids that were really the focus of her life until we lost her. So we explicitly found out about Huntington's disease when my mom started getting kind of subtly symptomatic. And I was about 19 or 20. I was in the American Army, so that's me on the left. That's me actually in Kosovo during peacekeeping rotation that I was there in 1999, 2000. While I was there, one of my jobs, I was kind of a communications guy in my deployed tank battalion. My job was to help set up the internet, which, you know, existed then, but was pretty, pretty rudimentary compared to what we had today, which is for better and worse. And so one of the things about being the internet guy is you always have the internet. So I was kind of obsessively trying to understand this Huntington's disease thing, you know, and we had sources like this. So people who've been around for a long time might remember this place, a place on the internet called the Huntington's Disease Lighthouse was available. This is sort of a screenshot from back then. And, you know, it was a great place for the community to get together, but it was a terrible place to learn about science. You know, as you can see, you know, Andrew Weil here is saying you can use diet to fix HD, which obviously is terrible advice. So I was really frustrated and I felt really annoyed, frankly. And I decided that when I got out of the Army in 2001, which was a great time to get out of the US Army, I decided that I would go back to college and at least take some biology classes so that I could understand what was happening with my mom and what would be happening to all of us. And so that happened, and I got out and I went to college and never looked back. I was very lucky strictly lucky to go to the University of British Columbia for my undergrad, which happens to be one of the best places in the world to do HD research, thanks largely to the efforts of Michael Hayden, who'd been there for decades and has an incredible both basic and clinical research program that I just sort of happened to be an undergrad at a school at. So basically, I joined Michael's lab and never looked back, and I've been working on HD ever since, and now in my own lab here in Seattle at UW. You know, of course, as a family member, I was incredibly motivated and moved by the Wexler family story, in particular, you know, Nancy's tireless efforts at wrangling and organizing a whole field of people and governments and funding agencies to work on HD, which at that time had essentially, you know, no recognition and no research happening and no hope. And the HDF has been an incredible, you know, inspiration for me. And, you know, my first academic conference I went to was an HDF meeting, and just incredibly pleased to be here and to be participating in the HDF research and to now have this great grant from the organization. So lots of love for the HDF. Of course, the key thing that we thought for all of that wrangling research that Nancy helped coalesce was an answer to every HD family member's question, which is, will I get symptoms? And in '93, of course, we discovered a means of trying to figure out if someone's going to develop HD or not. And so unlike in my grandma's generation and my mom's generation, where people was in the shadows and people didn't talk about it, now one could, if one wanted, you know, test and find out whether one had the mutation or not, thanks to this incredible work. And so, you know, I've gone through predictive testing and found that I unfortunately carry the mutation that my mom had, as do a couple of my siblings. So unfortunately, despite having this incredible resource of the gene, you know, the quest continues. So everybody's been stealing this slide as researchers from each other for so long that no one knows who actually made it. If you do, apologies, please tell me. But these are coronal sections of the brains of a person who didn't die with Huntington's on the left and someone who did on the right. And you can see that there's this really vast atrophy of this one particular brain region of the HD patient. And that's the fundamental question that motivates my lab, is how does one go from the left to the right by inheriting this one precisely defined and identified mutation? And I'll start with a quote. This is one of my science heroes. This is a guy named Sidney Brenner, who did get the Nobel Prize, but probably should have gotten 2 of them. but that's a whole other talk. Sydney was a very cantankerous, amazing guy. I got to have drinks with him one time before he passed away recently, which was a great honor and fun for me. And he has this great quote that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Meaning that as much as we like to think that it's our clever ideas, it really matters a lot to have new techniques. And the work we're trying to talk about definitely fits in that category. I won't go into any details about this, not least of which because I don't understand the mechanics entirely myself. But I'll just say that sequencing techniques that have emerged in the last couple of years are just totally insane. It was already crazy what we could do with what are now kind of traditional small short read sequencing techniques that were actually only evolved when I was in grad school. But this new generation of sequencers actually can sequence DNA or RNA at individual nucleotide level and in real time. So this technique is that I'm going to show you data for is from a company called PacBio, and their technology, it literally takes a video of DNA being sequenced and records the identity of the base in real time. It's just an absolutely bonkers advance in how we look at DNA and RNA and what we can do with it. A little bit of background. So our project is looking at splicing. So what is splicing? So most of our genes, we have thousands of genes in our genome, not just the Huntington gene. Almost all of them occur in these kind of chunks or chapters of code that are interspersed with DNA that's not part of the gene. And it seems weird until you get really deep in biology, and then it's amazingly beautiful. And there's a lot of reasons for it that we don't need to talk about. But basically, just realize that your genes are organized in these chunks, and that a lot of the genes in our genome are different chunks get used or included in the actual gene as it gets turned on at different times and in response to different cues. And because of the way they work, what are now the traditional sequencing methods are really terrible at figuring out which of these versions, because they only read short bits of the gene at a time. And so it's very difficult to reassemble it. And so one of the things that's been really markedly changed by the advance of these new sequencing technologies is that you can do much longer reads. And so you don't have to sort of infer what the organization of the chapters was. You can literally just read it on one big long stretch of DNA or RNA, which is incredible. So the work that's been going on, that's funded by the HDF, is really taking place largely in the lab of our collaborator, Paul Baumanis, shown here on his lab photo, and highlighting some of the folks in his lab that are really doing the work at the bench right now. And like a lot of things, this was kind of fortuitous. I saw a talk by Paul where he talked about this really interesting result they had, actually in Alzheimer's disease, where they had looked at this shuffling or splicing of the genes that when mutated cause a familial form of Alzheimer's disease. So most Alzheimer's is just bad luck, but there are families that have these rare mutations, and some of them have been seen here in Seattle by colleagues or by clinicians that folks might recognize, like Tom Bird, who's of course been a huge HD researcher in Seattle forever, and Sumi Jayadav, who's kind of taken over as the new Tom here in the HDSA Center of Excellence here. So this was their work in Alzheimer's disease that sparked us, and they found really interesting splicing changes in these other genes that cause Alzheimer's disease in these other families. And when I saw Paul's talk, I more or less said, that's an amazing technology. Like, we should really try to do that in HD, and he was up for it, luckily. It's fortuitous because Paul's kind of background interest, he's really interested in repeats in the genome, and since HD is one of those diseases, luckily it wasn't too hard to talk him into working on this project with us. The other thing that I'll mention just briefly is we're looking not just at HD brains, but another very rare disease that's only about 100 patients that have been described, called Huntington's disease like 2. This is one of a set of syndromes that are sort of, they're so reminiscent of HD. I mean, these patients look just like HD patients, even to the most trained neurologists. And even if you look at their brains while they're alive with an MRI, or after they pass away with microscope, the brains look exactly like HD patients. So there's something about these patients that's very much like HD in terms of how the disease manifests. But the gene that's mutated is a totally different gene. It's not the Huntington gene at all. And in fact, it's this gene called junctophyllin 3. And it's known that splicing is a part of this disease. And so it's very interesting to us that there's potential splice involvement in a disease that looks so much like Huntington's disease that you can't tell them apart. And so we decided to look at brains, not just of HD patients to see changes of splicing, but this HD-like2 as well. And so we worked with Russ Margolis, who's at Johns Hopkins, who's an HD researcher as well, who's mostly described and kind of led the world on this HD-like2 disease. And Russ's lab very kindly shared with us, as far as we know, the only brain samples from HD-like2 patients in the world that we're aware of. I should say one thing about HD-like2 is on top of being very rare, it's also almost entirely found in Africa. And so the patients in the US are mostly of African-American descent. And so most of the disease is found in Africa. And so it's really, unfortunately, understudied. So we were excited to be able to work on this as well. And so the basic question that we're trying to answer with the funding kindly provided by the HDF is to use these new sequencing techniques to sequence the RNA of the Huntington gene at incredible depth, like tens of thousands of times for each molecule, so that we can get unprecedented accuracy. And so if your sequencing is not accurate and you see something weird, like a change in a nucleotide, you can't really be sure if it's just noise or whatever. But with these hyper accurate sequencing methods, you can be really, really sure that what you're seeing is real. And that lets you look for the exceptional things like small changes in splicing patterns, or even you can actually see changes to the RNA sequence itself in a really weird process called RNA editing. After it gets made, some RNAs are actually edited. And so that's hard to see in the noise of normal sequencing. So we're really interested to look at that. And because we're doing this compare and contrast thing, we're looking at the Huntington gene itself, of course, but also this junctophyllin 3 gene, which is the gene that, when mutated with a CAG expansion, causes this HD-like disease. And it's by comparing and contrasting similar but different diseases and different conditions that you can really learn a lot in biology. And so this is just a text slide for the aficionados, but what were we hoping to find? We were looking to see if we can see expansions at the CAG at the RNA level. Can we see evidence of a particular form of the Huntington gene that Jill Bates's lab has done really beautiful work describing the impact of? Do we see alternate splicing of HD, shuffling of those chapters, which nobody's actually reported before, which is really weird because HD has 67 chapters and you'd expect them to get shuffled. And can we see these editing changes to the actual post translational transcriptional changes to the RNA sequence itself? And then, of course, to us, a really interesting comparison is what do we see in HD-like2? So very tentatively, and I'm going with the HDF way of kind of like showing unpublished and very much in process data. The initial runs that we got from the HD are very surprising, and we found this very interesting phenomena that now I have to learn about because it's new to me, which is a very extreme form of change splicing, which is actually circular RNA. And so we found little circles of the Huntington gene because the splicing that happened didn't go from chapter 1 to chapter 2, and went from chapter 2 back to chapter 1 and loops, and actually literally loops back in on itself and forms a circular piece of RNA. So we found this in the brains of HD patients, and we're really interested in understanding how it emerges and what it means. And of course, again, we really want to look at the HD-like2 brains as well. That's been kind of interesting. So the Institutional Review Board at Johns Hopkins wanted us to get consent from the donors of the brain tissue, but who had unfortunately, of course, passed away because they had donated their brains. So sometimes that's the kind of thing you have to deal with as a scientist. But we've, with Russ Margolis's help, we've overcome that, and the HD-like2 brains actually just this week arrived here in Seattle. So that's just a little quick snapshot of where we are. I'd like to thank our collaborators in the Valmanis lab who are really driving this forward, and Russ Margolis, who's given us access to the HD-like2 brains, and of course to the HDF for generously letting us apply these new exciting techniques and really trying to do some kind of blue sky research, which is so hard to get funded these days. So we really appreciate that as well. And with that, I'll stop. Thanks.
Thanks so much to all you guys for sharing your projects with us. It's super cool to see people studying HD from such different aspects. So we really appreciate you sharing your work. And thank you to everyone who's been submitting your questions. Keep submitting those throughout the Q&A, and we will try to get to as many of those as we can. Again, whatever question you might have for our speakers. So I think I will start with one for Rachel. This was an anonymous attendee asked if the conformation and the charge of the Huntington protein is the same between wild type and the mutant form.
Yeah, so there is definitely evidence that this really big Huntington protein molecule might change shape when we have it in the either disease form or the regular form. But it really seems to depend what else it's hanging on to. So one of the partners that we know a lot about for Huntington is this other protein called HAP40, because scientists have really unimaginative names for proteins. And basically, when Huntington is together with that, what's really interesting is that we don't actually really see many differences. And this is almost like it's even more, makes me even more interested, because there's actually not many big changes that happen between the regular and the disease forms of Huntington. But the thing that's important to remember is that picture I showed you earlier of what the Huntington protein looks like. That's just all the really rigid parts of the protein that we can see in the microscope very well. There's about 25% of the protein that's missing, and we don't really know what they look like. And it's what's really cool is we've got all these AI guided, sort of simulations that we can do now and all sorts of bits and pieces. So this is something that loads of people are looking into to try and figure out what all those other parts of the protein look like when we have their expansion, and see how they might differ between the two forms of the protein.
Thanks, Rachel. I will pass one to Tam now. So this is from Nick Karen. He says, "Hi, Tam, cool talk. When looking at PAR and CSF, did you look at the relationship in pre-manifest group with the years to predicted onset, or the relationship of PAR with disease burden in all mutation carriers?"
Hi, Nick. Yes, we did look at whether PAR levels correlated with disease measures, and they don't. So what this means is, because we were hoping that, PAR might make a good biomarker. And what that means is it wouldn't make a good biomarker. There's lots of things that can affect, at the moment that you're taking that sample, is the person sick that day, or, do they have an immune response? Or sometimes things don't track so nicely with disease measures, and they don't make a good biomarker. So the answer is it doesn't track with disease, but there's just this robust decrease in both pre-manifest and manifest patients.
Thanks, Tam. So here's one for Jeff from Ricky Hirschhorn. Are the branch points of the circulate RNA determined yet? Are they all the same?
When Paul and I last talked, we had just found one that was identified, and I'm not going to say the exons, not because I'm hiding them, because I don't remember. But yeah, the one that had been most abundantly identified is identified, and it seemed clear at that time that it was mostly this one species, as far as I remember. And actually, Paul's might be online. He could chime into the chat and tell me if that's incorrect. But yeah, it seems like we definitely have identified the branch points.
Thanks, Jeff. So going back to Rachel, also from Ricky Hirschhorn, what does NEAT1 encode, and do we know its function?
Yeah, sure. So NEAT1 is what we call a long non-coding RNA. So this means that it's an RNA message molecule that doesn't actually get turned into a protein. The RNA molecule itself is kind of the end product. And this is NEAT1 plays this really important role in structures in the nucleus, which are called paraspeckles. So these are kind of big globules of proteins and RNAs that all kind of stick together. And they have really important roles in the way that cells deal with stress responses. If something bad happens, the cell needs to rapidly respond. Paraspeckles are quite important in that. They also, play a role in different processes to work out which genes get switched on and off, and other bits and pieces. So they're very important for the health of cells. So this is really interesting because we know Huntington itself is also involved in stress responses. And so we're doing more work on that. Well, I say we, Manisha, who's the PhD student, is doing a lot more work on that because she's much better at this stuff than me, and trying to figure out like how this is all working and how it's all fitting together.
Thanks, Rachel. So for Tam, this is just a comment from Hannah Beasley. She says, it's really cool how you're looking at approved drugs to treat HD. So maybe that's something we could hear about if we have you back, and you could talk about that. And then also from Wendy Welsh, she asks, I would indeed be interested to know a bit more about how DNA damage is measured in small blood samples.
Yeah, sure. Kind of the gold standard is something called a COMET assay, where you basically have your cells and you run an electric current through them, and if the DNA is broken, it spreads out and makes this sort of comet-like shape. So you have the nucleus looks like the head of the comet, and there's a tail. So my original plan was to use a very high throughput automated way of using the COMET assay. I've done lots of COMET assays, but they're very clunky and you got to like put some gel on a slide and all of this stuff. Not something that would be great for, a clinical lab to be able to run lots of samples. And so what I was hoping to use was this commercially available, 96 samples you can do at once, and it's all lined up beautifully for the microscope and so on. But that product was discontinued for some reason, and I'm not interested in trying to generate a protocol that can't be used widely. So I've pivoted now to the two approaches that I'm looking at are qPCR. So I don't know how familiar everybody is with these techniques, but it's a way of measuring, on a long piece of DNA, how many damage sites there are. And then the other way is actually something that's being developed by Carlos, a PhD student in our lab. And it's called a RAD assay, which stands for Repair Assisted Damage Detection. And this is cool too, because it's a way of using microscopy to look at direct DNA breaks. So you've got the damage there. You use enzymes to fill in the damaged bits with labeled nucleotides. Sorry if it's too technical. And then you can measure how many spots got filled in by fluorescence on a microscope. And we have seen in cells from HD patients, you can get really specific with this, with what types of damage it might be. Is it a base damage? Is it a break? Is it a break in one strand or two strands? And we can see that in cells from HD patients, we see very distinct differences from cells from control patients this way. So I'm trying to adapt that to whole blood samples and basically getting them to stick to the slides is is a challenge, but I've got a number of approaches that I'm working on. So I hope that answered your question.
Thanks, Tam. And going back to Jeff, and I think it was Ricky who asked this question about what the exons were, but Paul answered in the chat, exons 6 to 2. And we also have a question for you, Jeff, in the chat from Manganesh Kalogay. I'm sorry, I'm sure I butchered that. But they asked if you could enlighten more on splicing events and expanded repeat diseases and if it differs from normal splicing.
Yeah, what's really weird about Huntington is that, as far as I know, and someone could correct me if I'm wrong, it's not alternatively spliced. So it has 67 exons, 90-something% of human genes that are multi-exons are alternately spliced. And Huntington is one of the ones that, as far as we know in the literature, isn't. And that's one of the reasons we want to look at it. There's some speculative new papers about CAG repeats themselves becoming part of the splice machinery. That's kind of all new. But as far as I know, it's what we're looking at is kind of like standard alternate splicing at the genes that we're looking at. We're set up to see surprises like these circular RNAs, but our hypothesis is not that there's something distinct splicing wise happening at these genes compared to splicing across the genome otherwise at other genes.
Thanks, Jeff. So this one may be for Tam, but I'm sure other people could chime in. It's from Igor. He says, thank you for your contributions. Amazing work. Now there is information on how somatic expansion is not the single element that causes the disease. What does this mean to companies developing treatments for somatic expansion?
Yeah, so I think I don't think anybody would say that somatic expansion is like the single cause for disease. I think that we have very, very strong genetic evidence that it's causative. And I'm really glad that there are companies working on targeting that aspect. I think it's just the idea of there's probably more to the picture than just this one thing. And so, for example, what if global DNA damage is what's driving this somatic instability? then that makes global DNA damage a good target as well. So I hope that answered your question. Did you guys have anything to add?
No, I think you did great.
So this, maybe we'll end with this one, and I think this could go to each of you, and you might each have kind of something additional to add. So Brianna Esker asks, what do we think the actual purpose of the Huntington protein is? The age-old question.
I can maybe start, because I think it's crazy to me that we don't know. It just bothers the hell out of me. And you know, it's one of the things my lab's working on. We study that in part by looking at what happens when you knock out Huntington, which people haven't done as much work as I wish we would have done as a field on what happens when Huntington's gone, because that can kind of tell you what it was doing. So we have a lot of work on that. All I can do is like agree with the frustration, and it's crazy to me that we've gotten this far and trying to lower Huntington, for example, therapeutically, which I'm glad that we're doing, without having a good answer to that question.
Yeah, maybe I can jump in next. So I always like to think of Huntington as this cellular Swiss Army knife that does like all of these different jobs. And it does all those things by partnering with lots of different molecules in the cell. But what's still very confusing is that we've spent a lot of time in the field of Huntington's disease generating more and more lists of everything that Huntington interacts with, but not actually really following them up or validating them or understanding what the functional consequences of those are. So currently, I think it's over 3,500 different protein molecules are reported to be interactors of Huntington, but we only really have structural evidence for one. So that tells me that either we're not very good at making these lists, or actually it's very, very complicated. Yeah, so that's also what my group is trying to figure out as well.
Yeah, and I would just reiterate what both of you are saying. It's very frustrating to not know. It's not that we don't know what the Huntington protein does. We know lots and lots of things that it does. We just don't know exactly which ones are important in terms of disease. And I think that we are a little bit, we may have been led down some garden paths because of some of the model systems that we used for so long. It's really hard to figure out what 3,000 amino acid protein does when you only look at the first 80 amino acids, but it's not an easy protein to work with, as Rachel will attest. And so for a long time, people really just used just the little tiny bit that is expanded in HD. But that, so I think we've been led down a lot of garden paths by our model systems, but I think that's changing. A lot of labs are really looking more at what we call the endogenous protein, which is the one that exists in nature, and not ones that we fabricate ourselves. And we're also looking a lot more at human data. We need mice to test certain things, but the best answers are going to come from human samples, and a lot more labs are focusing on that too. So, yeah.
Great point to end on, Tam. Thank you. So we are at the end of our time, so I'm going to wrap it up there. If you had a question that we didn't get to, I will send all of those to our speakers. So if they are willing, they can follow up with you directly. I want to end by again thanking our sponsors, Neurocrine Biosciences and Wave Life Sciences, as well as everyone who donates to the HDF to support the research that we fund. Of course, our audience, and last but certainly not least, our speakers. Thank you all so much for being here. Our next webinar is Tuesday, April 23rd, and will feature the work of one of our 2023 Transformative Research Award winners. We will be joined by Drs. Bev Davidson, Leslie Thompson, and Jang-Ho Cha, who will tell us about their work using gene therapy for Huntington's disease. So thank you all so much for joining this webinar, and we hope to see you next month.